So today we're going to finish up 10.3 gene pools and speciation. And the questions to consider that we're going to look at are we're going to be looking at there's actually a couple of different modes of natural selection. So natural selection doesn't always happen in one direction, like we've often looked at in our example. So we'll take a look at what stabilizing and disruptive selection are. We're also going to look at speciation in depth and how do we go from one species to a couple of different species. And then finally, we're going to look at the idea of how the tempo of speciation, how quickly a species can evolve into two new species. So let's take a look at our PowerPoint, and we're going to start with the modes of natural selection. I can get it to pop up there. Okay, so modes of natural selection. So if we have an original population with continuous variation, right? So this maybe was caused by polyploidy or something like that. So here we have continuous variation in mice. So they range from very light color to a very dark color. Most of the examples of natural selection we tend to look at are directional. So where one of the phenotypic ends of that population is selected for and the other one is selected against. So in this case, we see the mice that are being selected for in one direction. This would be similar to like the peppered moths, right? So either the dark color is selected for and the peppered one is selected against, or the other way around. So when we start to see one of the extreme phenotypes being selected for and the other ones being selected against, then we'll see directional selection. But we also saw an example of disruptive selection. When we were looking at that rock pocket mouse, there was this habitat that there's lighter rocks and then there was the dark black rocks. Well, if you have a population that is, has this original population having a gradual change in phenotypes, like a range, but then the habitat is kind of one or the other, that can cause selection for two extremes. So the dark mice were surviving in the dark black lava rocks, where the lighter mice were surviving in the other portions. But all of the intermediate phenotypes were being selected against. So this is disruptive selection, when you are selecting for the two extremes of a phenotypic variation and the intermediate types are selected against, because these guys could be seen on either the dark rocks or the lighter sand color. Stabilizing selection, on the other hand, is when the intermediate phenotypes are actually being selected for. And a classic example of stabilizing selection is baby's birth weight. I apologize, this is in pounds, but it was like the neatest picture I could find that wasn't so blurry. But you can notice that baby's birth weights tend to center here, kind of around the 7-pound area. And that's, about 3 kilos more or less, like 3.1 kilos or something. And this other line is showing you the mortality. So you can see that babies that are really big and really small tend to have a higher mortality. They're selected against. This mortality is often has a lot to do with the fact that a smaller baby might be underdeveloped. It's going to, die more frequently. Maybe it was born super early. The larger babies might have issues with, if they're so big, it might actually kill the mother in childbirth more often than a normal-sized baby would. And it could also have to do with like gestational diabetes type issues, so that the offspring might be more prone to things like that. So then we have this optimal birth weight. So more babies are selected for right in this middle range. Okay. So that's our classic example of stabilizing selection. So you can see the 3 types of selection are directional, disruptive, and stabilizing. Okay. Now we're going to look at speciation. And speciation is when you go from, one species to 2 species. And there's a couple different types of speciation we're going to look at. We're going to look at temporal, behavioral, or geographic. So how do they become new species based on some timing stuff, based on different behaviors, and then based on geographic isolation. Then lastly, we're going to look at this one other type called polyploidy that's going to give us an idea about what happens when we start to get non-disjunction, causing a whole new number of chromosomes. All right. So the definition of species: it's a group of organisms that can interbreed and produce fertile offspring. If for some reason isolation occurs where they can no longer, like if you got two groups of that population, if that population gets split somehow, whether it's through habitat, geographic isolation, some temporal or behavioral matters, if that population gets split, then if there's different selective pressures acting on those two sets of the population, then it could result in enough genetic changes that if you were to introduce those two parts of the population again, they could no longer interbreed. And if they can't interbreed anymore, they're no longer the same species. So if they're isolated from each other, there might be different environmental pressures working on them, so they're going to evolve differently. You get enough changes in the gene pool, and if those two sets of populations can't interbreed, we have a new species. Okay, so geographic isolation is probably the easiest one to think about. Geographic isolation is when there's literally a physical separation between two populations within the species. This example can be exemplified in the tree iguana from South America, which was on the mainland in the jungles. And they think that a small population of those iguanas ended up riding a raft across the ocean and landed on the Galapagos Islands. Now, on the Galapagos Islands, it's a whole different landscape. It doesn't have all these tall tropical trees and all the different fruits that iguanas would normally eat. But there is a lot of algae, and that algae, kind of in the ocean that they could get to, that started causing a different selective pressure over time. So they're in two different locations. They can't interbreed with each other because they're so separated. The selective pressures here for this iguana were, can you swim? Can you get down and get that algae to get food? Because there wasn't the same trees and other things that there were in the original population. So in this case, that geographic isolation is causing enough genetic changes because of different selective pressures over time that those two populations are now separate species altogether. Habitat isolation is essentially a type of geographic isolation as well. So it can kind of happen in a closer area. This is showing you two species of garter snakes, one that tends to live and hunt and be in the water more often, where this one tends to be on land. And so it was thought that they did share a common ancestor, that maybe some individuals were really starting to exploit the water more, where others were staying up on land. And because they weren't interacting as often, it ended up dividing into two different species. Okay, temporal isolation. This is isolation that can happen if major events, especially like breeding seasons, happen at different times for certain populations. And this is the eastern and western spotted skunks. Well, the eastern spotted skunk mates in the wintertime and the western mates in the summer. So even if those two populations were together anymore, they're no longer going to be able to have offspring because they're not mating at the same time. So those two populations will no longer mix. So temporal isolation could be if they're breeding at different times of day, different seasons, different years, and their gametes just aren't going to mix because maybe the females are not in estrus or not, fertile at that moment. And behavioral isolation is when certain parts of the population might start to develop courtship rituals that are really important. And courtship rituals in birds are a pretty big deal. What they show, they say, hey, I'm the same species as you. I'm not a threat. I'm not trying to hunt you. So I'm going to show you this cool pattern of stuff so you know that I am okay. I'm not a threat. And that whole dance, I really like this one, of the blue-footed booby dance, is required for birds to be able to pair. And if they don't, they're not going to pair with, say, the yellow-footed boobies because they do not do the same dance, don't have the same high step and other things. And so you don't see a mixing of gametes because they're not going to be pairing with anybody who doesn't do that dance. There's a really nice video that I'll put on the link that you can see of the blue-footed booby doing their mating dance. It's pretty cool. Okay, so we looked at behavioral isolation, temporal isolation, and geographic isolation. There's also something can happen at the genetic level that can cause speciation. So it's almost sort of an instant speciation, is when non-disjunction occurs And if non-disjunction occurs and the offspring actually end up being quite viable, then they can kind of become a whole new species because they don't have the same diploid number anymore. Maybe they have a triploid number or a quadruploidy in this example. Now, this can happen pretty frequently in plants, and you're going to get to know an allium example when you're doing biozones. That's the example that you really need to know, is the one about allium. That's onions. But when you get polyploidy happening, the new offspring can no longer mate with the original population because they don't have the right set of chromosomes. But with plants, because they can clone themselves, with shoots and runners, that they can actually then cross-fertilize with the new plants. So that were clones of themselves. So let's say you have an allium plant that all of a sudden has a different number. It's no longer a diploid plant. Maybe it's a quadruploidy plant. Well, if that quadruploidy plant can clone itself and make new ones, then the new individuals, those clones, can actually then crossbreed with each other. And so that's creating this whole new species versus the original species that had a different set of chromosomes. So it's most common in plants, but there are some examples of animals like the red viscacha. It has a diploid number of 102, and they think that that's because of some sort of polyploidy event resulting in speciation. A really interesting example of speciation, though, has to do with this mutant crayfish. That's actually a really terrible invasive species taking over Europe. It's originally from like Georgia and Florida in the United States. It was a slough crayfish, and they think that one of these, I call them crawdads where I'm from, so they think one of these crayfish, crawdads, not a non-disjunction event happened in either the mom or the dad, and so the egg was diploid, and then the sperm was haploid. They came together and resulted in a viable triploid crawdad. And this viable triploid crawdad could no longer reproduce with the diploid crawdads. But for some reason, something about the mutation, it was able to clone itself. And then because it could clone itself and make a bunch of eggs that became viable, triploid viable eggs, then all of the offspring were clones of the mom, right? And now it was sold as a pet in Europe, and because it didn't need to have a mate, so somebody would get like a crayfish and put it in a tank, and it would start making new babies, and then they would just like release those babies into the wild because they didn't know what to do with them or flush the eggs out, and now the population has just gone totally crazy. The scientific name, now that it's been named a new species, is Procambarus virginalis. Okay. Now, you might see this when you go to university. I don't think the IB uses this terminology, but this is a common way to describe speciation. There's allopatric speciation, when you actually have physical separation between the two species resulting in speciation. And then there's something called sympatric speciation, where it can result in not an actual physical barrier isolation. So that could be with a temporal or definitely with alloploidy. So sympatric speciation is when it happens when they're still in the same location. Allopatric speciation is when they happen in two separate geographically isolated places. Again, I don't think that IB uses those terms, but they are pretty common when you're studying evolutionary biology. Okay, very last thing, tempo of speciation. So originally, the main thought was that evolution always just happened gradually. So those changes started to evolve very slowly over time. So you see this species, and maybe we saw some speciation happen with a few genes, and then as time goes by, this one starts to get gradual changes, and then this one starts to get gradual changes, and then they're definitely two different species. Well, that does happen. Gradual speciation does happen, but there was also a lot of species that they were finding really big gaps in the fossil record. And some dinosaurs and other things, and a lot of mammals. Well, it turns out that you can see these big gaps in the fossil record without these intermediate forms, with this type of pattern called punctuated equilibrium. So the punctuated pattern is when it's the same environment for a long period of time, and then boom, there's some dramatic environmental change. And then that might cause two forms to be selected for that are, two extremes of something to be selected for. And then you start to see that change, no change or not as much change over time, because if there's periods where the environment's stable, there isn't really a need for selective pressure to happen. So you might see very slow or very little change in between. So punctuated patterns shows long periods of stasis, same, a rapid evolution due to some environmental event, and then you might see more stasis again. So it's like this long gradualism with these punctuated patterns in between. Some examples of punctuated equilibrium result in something called adaptive radiation. So adaptive radiation is when you have one species and then boom, 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 a bunch of new species evolve pretty rapidly from that one original parent species. That happened on the Galapagos Islands with the birds. We had one species of birds coming down and landing on the islands, and then it was populating all these different islands, and on the islands, they were pretty new. There was a whole bunch of different niches available because there weren't other birds living there at the time or taking advantage of it. And so because there was a whole bunch of different things to eat, different pockets of these birds started specializing in different food types. And on those different islands and in different places, there was different selective pressures. And we started to see this rapid evolution of all of these different birds. This adaptive radiation also happened with mammals. So mammals were just a few different species. So this is showing actually, the width of this is showing how many different species exist. And there were only a few different species of, or like families of mammals living up until, we saw them kind of split off into the monotremes and then into the marsupials and the placental mammals. And there's still not a ton of families. We saw another split around 140 million years ago, and this is likely when Laurasia became, Australia becomes isolated from a lot of the rest of the world. And then the placental mammals are in the, most of the other continents, Asia, Europe, Africa. Now, not a lot. Then all of a sudden, about 65 million years ago, we saw this huge explosion. So this line is thicker, meaning there's a lot more families. Well, that happened because the dinosaurs went extinct. Because before, the dinosaurs occupied all of the niches available on the planet. But when the dinosaurs went extinct, all of a sudden there was not a lot of competition to fill all the niches. So when the plants started to recover, the mammals that had survived, they started to take advantage of all those niches, and we saw a really fast, rapid evolution of mammals. And that's now we're in the Cenozoic era, which is dominated by mammals altogether. And that is adaptive radiation of mammals that happened after the niches became available when the dinosaurs went extinct. Okay, so that is the end of this section on 10.3, and there's a couple other videos that you can kind of see some examples.